So we'll now move on to the genomic medicine theme, and I have the great pleasure of introducing Dr. Amy McTague, who is a clinician scientist. She has a growing group at the Zayed Center for Research into Rare Diseases in Children. She is also the deputy lead of the BRC's genomic medicine theme, and Amy is also an honorary consultant pediatric neurologist at Great Ormond Street. Thank you, Amy.
Okay, hello everybody. It's a great pleasure to be here to represent the genomic medicine theme. So as Kiki said, I'm the Deputy Theme Lead Clinical, and it's a pleasure to present an update on behalf of our lead, Lynn Chitty, Sergio Castellano, and our junior faculty and early career researcher co-leads, and we're supported admirably by Dowda. So our theme really works at the cutting edge of genomic science, and we work in a number of different disease areas, which we'll highlight to you here. So the first of these is increasing the speed and the yield of genomic diagnosis, going from the fetal period across childhood really, and bringing those cutting edge technologies into the clinic. Then we have a large number of projects working on exploring applications of cell-free DNA, where we're able to use this free-floating DNA within body fluids rather than within cells to improve diagnosis and to understand disease mechanisms. Then we'll hear about work in pathogen genomics, and lastly about work in understanding rare disease at a single cell and spatial resolution. So one of the projects supported within the genomic medicine theme is Gene Steps, which is a project investigating the genetic causes of early onset epilepsy under 12 months of age. And this is an exemplar project from an international precision child health collaboration between Toronto, Boston, Melbourne and ourselves. This has been supported by the BRC and subsequently by Gosh Charity, and through this we've been able to gather a really large cohort of babies with epilepsy, over 120 now at Gosh, and over 500 internationally. In this group we've proven the individual and personal clinical utility of rapid genetic sequencing, returning results within several weeks in over half of the infants. We published our pilot cohort, and we're now moving towards solving the unsolved cases using cutting-edge genomic techniques, working on multi-omic techniques, and through a collaboration with the Child Health Informatics theme, looking at using health economic analysis using EPIC data. We're also now starting to think about not just diagnosis, but treatment and how early we can stratify these babies towards personalised and tailored medicine approaches. Rapid sequencing has also been a theme in fetal samples. So the genomic medicine team have led on the use of fetal exome sequencing, receiving a large number of national referrals with a rapid turnaround time and a very high diagnostic yield. This has now been translated into clinical service within the NHS, and a network of excellence has been developed with an in-house rapid pipeline. The team are now moving towards, again, bringing those cutting-edge techniques into the clinical service by evaluating their yield, but importantly, their impact and their acceptability to patients and families. We've also looked at a metagenomics, which is a cutting-edge genomic technique that allows for the identification of bacteria and viruses from samples from patients, many different types of samples. And this is untargeted, meaning it can look for an infection without having any kind of prior presumption about what the infection may be, which is a real departure from the usual methods. This pathogen agnostic approach is very useful, for example, the surveillance of outbreaks of disease. In the bottom left of the slide, you can see when there was the global surge in childhood hepatitis cases, which Judy Brewer and her team were able to use this technique to identify the cause of this, which was co-infection with AAV2. And again, at the bottom of the slide, you can see a photograph of a young lady who had an occult undiagnosed eye infection, which nearly led her to lose her sight, but after a 2-week course of tailored antibiotics, was able to restore her vision and live a normal life. So this is finding hidden infections and really improving care for patients. This is now the first and only nationally accredited service using metagenomics to diagnose infection. Indeed, we are also not only using rapid techniques and new techniques, but in this part of the theme, we're going from cells to looking at organoids, as we've been hearing earlier today, and then using these to also assess the generation of organs. In this one example, we're using spatial transcriptomics, which is a development of single-cell sequencing. And this allows the exploration of not only what genes are doing within cells, but then what genes are doing within cells within their spatial location within tissue. And in this example from work from Serena Brown, Manju Kurian, Sergio Castellano and George Hall, they've used this to really analyse how well an in vitro model can recapitulate brain development, in this case of the midbrain, to understand patterns of brain development in severe childhood neurological disorders. This is really key to implementing these novel in vitro models. So in summary, in the theme, we're harnessing these advanced genomic techniques and transcriptomic techniques to bring them really from the lab and to understand what they mean for our patients and to bring them into the clinic. And at the same time, we need to really understand what the impact of these accelerated and novel diagnoses are for our patients and their families, using qualitative research, using health economic research. We're well integrated across the themes, particularly with the novel therapies themes, because we're able to not only generate targets for novel therapies, but also to understand the impact and side effects of these therapies. And of course, using data science and EPIC to measure the impact of these is also important. Lastly, our major focus is developing the next generation of genomic scientists who can really understand and implement these novel technologies. So that being said, we'd now like to introduce Flavia and Ben to show two really exciting projects in the theme.
Hi everyone, my name is Flavia. I'm one of the BRC's funded PhD students, and today I'll be speaking to you about Mosaic Brain, which is a new diagnostic test that we're developing for patients with focal epilepsies. And the reason why this work is important is because 30% of children with epilepsies still do not respond to the current treatments. So having a better understanding of what causes the seizures in these children help us provide a better care for them. With that in mind, the main goal of this work is to increase the number of genetic diagnoses in children with focal epilepsies. And this is because having a genetic diagnosis may help with finding personalized treatments for these children, also support better patient care, namely with helping us find out if there would be further tests or monitoring that the patients would benefit from, but also will help us help families with their family planning by understanding what is the risk of another child having epilepsy as well. So during the initial phases of the process of planning for the project, we had a lot of support from several patient and public involvement and engagement groups, including our very own Gosh Young Persons Advisory Group, for which I'm very thankful because they have given us invaluable feedback and a lot of support to get this work off the ground. And because of them and their support, we are now able to work with a group of children with focal epilepsies. Just to give you a little bit of context, focal epilepsy is a specific type of epilepsy where children have an affected part of their brains that causes their seizures. Some of these children are able to have an operation to remove the affected region of the brain, and this is to help treat or even stop their seizures. So currently what we're doing in the project is we're then taking DNA from that brain that is removed on an operation, and we are going in to see if there's any changes in the DNA that would help explain the seizures in the children. You may be asking yourselves right now, why do we need to test the DNA from the brain and we can't do genetic testing just from the blood? And the reason is that for some of these children, the changes in the DNA that cause the seizures are not inherited from mom and dad, but they're new. And depending on when the DNA occurs, the change in the DNA occurs, for example, if it happens after fertilization, and particularly after the cell multiplication process has started, it's possible that the changes in the DNA are only present in the brain or even in the small part of the brain that's causing the seizures for the child. This is why it's important that we get that specific DNA so that we can test. But not all children are able to have this operation, so we don't have access to brain tissue from everyone. And because of that, we are now also working to improve our tests so that we can use it on cell-free DNA. As Amy mentioned already, cell-free DNA is a DNA that has been released from cells ultimately, and it's present in biofluids. In our specific case, we're collecting fluid that surrounds the brain, which is a less invasive procedure than having an operation. And that would allow us to extract the DNA that's present in the fluid, compare the DNA changes with the changes we're finding in the tissue to see if we could offer this test as well. So far, we've been able to test brain tissue from 25 different patients. We have found DNA changes that cause seizures in 11 of them. For 9 of them, there are targeted treatments that are available. Further testing has been recommended for 4 patients, and we have also helped one family by supporting them with family planning. We have gotten some very positive feedback from clinicians and families so far, And also in terms of our cell-free DNA, we've collected 12 samples from some of our patients, and we have their cell-free DNA already, and the initial genetic testing is ongoing on these samples, which is quite exciting. So moving forward, we aim to keep collecting samples so we can keep diagnosing more patients, improve the test to use cell-free DNA as an alternative, and for that, we will need to compare the DNA changes that we find in the cell-free DNA and in the brain tissue, so that ultimately, hopefully, we'll be able to offer the test just using cell-free so that we can test patients for whom we don't have their brain tissue. Thank you.
Hi everyone, so I'm Ben Paternoster. I'm a pre-registration scientist based in the NHS North Thames Genomic Laboratory Hub, and also doing a part-time BRC-funded PhD looking at using targeted long-read sequencing and hopefully using it to increase genetic diagnoses. So yeah, I've already kind of explained what the objective of my project was, but the reason this is important is because a confirmed genetic diagnosis can help the family plan for the future care and needs of the child. It also allows the exploration of possible treatment options, and also helps the parents plan for future pregnancies in terms of working out if it's more likely to happen again or if they have another kid with the same condition. So as very sort of a quick background, this is probably familiar to a lot of people, but obviously people inherit one copy of a gene from the mother and one copy of the gene from the father. And for some genetic diseases, it only takes one abnormal copy to be affected by the disorder, which is referred to as dominant. And for other genetic diseases, both copies need to be abnormal to cause the disease. And these are called recessive. So for the diagnosis of recessive diseases, we really need to know that both copies of the gene have been affected for us to give the patient a sort of proper diagnosis. So the sequencing we use in the NHS at the moment is called short-read sequencing. So if you imagine a person's genome is a book, short-read sequencing splits the book up into small sections, like a few words at a time. So this gives us small pieces of the puzzle, but it's often really difficult to figure out how it all fits together. So in DNA terms, if you look at here, we can see individual genetic changes, but we're often unable to tell how these changes are linked together. So in this case, this is showing what sort of diagram what short-read sequencing looks like. We've tested a patient and both parents. We can tell that one of the variants, the one on the left-hand side in blue, was inherited from the father, but the other change has happened spontaneously during development. This is called de novo. So we aren't really able to give a proper diagnosis unless we know it's happened on the copy of the gene inherited from the mother or the copy of the gene that's been inherited from the father. So the alternative technology to this is long-read sequencing. So with this, we get much longer stretches of DNA. So if I go back to the book analogy, with this technology, we're seeing full paragraphs at a time. So you can see how that's much easier to fit together. So it gives us a fuller picture of what's going on. So here's an example. So we've got longer reads that span larger regions of the genome, and using this, we're able to work out that this other variant that was detected on short-read sequencing has actually happened on the other copy of the gene. So that way we know that both copies of the gene have been affected, and then we can give this patient a proper diagnosis. So long-read sequencing is quite expensive, particularly if you're using it to sequence a whole genome. So my project is specifically looking at targeted long-read sequencing to look at just the specific regions of the genome that we're interested in. And by doing that, we should be able to reduce costs. So I'm going to show just a couple of examples. I've done quite a few cases now, but these are just a couple of examples which I think show quite the significant benefit of targeted long-read sequencing. So the first example was a prenatal case. So there was fetal imaging or an ultrasound carried out on a fetus, and they detected abnormal fluid buildup. So short-read sequencing was carried out on the fetus and the father, and there were two genetic changes that were identified in the fetus. One had been inherited from the father, but the other was actually unknown because the fetus was conceived using an egg donor, so we didn't have maternal DNA to test on. So this is quite a difficult diagnosis to make without that maternal DNA, but by carrying out targeted long-read sequencing, I was able to identify that the other genetic change had occurred on a copy of the gene that would have been inherited from the egg donor, which means that both copies of the gene have been affected, and it confirms the diagnosis in the fetus. In another example, this is in epilepsy. I believe it actually might be one of the GeneSteps cases that Amy was talking about earlier. There was a 1-year-old girl with seizures, decreased muscle tone, and developmental delay. So short-read sequencing of this girl and both parents revealed a DNA change that was inherited from the mother, which you can see in pink, And there's also another change that was not inherited from either parent. So again, this is what we call a de novo variant. So again, to make a diagnosis in this case, we need to know that both copies of the gene have been affected. So by using long-read sequencing, I was able to see that this new genetic change, or de novo change, had occurred on the other copy of the gene. So therefore, both copies had been affected, and we were able to confirm the diagnosis in this child. So looking ahead, long-read sequencing definitely has the potential to remove some uncertainty from genetic testing results and ultimately increase the diagnostic rate. And particularly targeted long-read sequencing is able to offer these benefits at a much reduced cost. So the next steps going ahead are to sort of scale up testing and move into clinical service. And yeah, thanks for your time.
Is that? Yes, hugely important area. Thank you. Questions? Are there any questions in the room? Online?
Flavia, can you give us a bit more detail on how you involved YPAG and children with focal epilepsy in shaping your research?
So I've been very fortunate also with Amy's help that we have had access to several groups of people, patients, families, carers for people that have focal epilepsy, but definitely the feedback from YPAG has also been invaluable. So in the beginning of the process of designing the project, we needed to figure out how we were going to present the project to prospective participants, and having the feedback from patients themselves, from families of patients, has allowed us to make our documentation, our presentation of the work to the patients much more clearer. So it gives us a little bit of confidence that the patients are making very well-informed decisions before they decide to participate in the study.
Thank you. Other questions? Thomas?
Yeah, just follow-on question to you. Of how many, well, what's the percentage of somatic mosaicism you're detecting from the resected areas? And is there something like a lower threshold of somatic changes which you have to respect? And how would you expect to find that back in cell-free DNA?
That's very good questions. Thank you very much. I appreciate that. So currently, our diagnostic yield is 44%, which is in line with what has been published before. We do think, since new publications have come out, that it's possible to increase the diagnostic yield if we are able to lower the limit of detection of the current tests. So that's the next step for the project, is to also be able to lower that limit of detection. Currently, we are able to detect somatic variants down to 1% in the sample, which is quite good already, but not good enough. We aim for more. I will say that there will be challenging with cell-free DNA. The current pipelines that we're testing use UMIs. So just a little bit of background. That's a way to identify uniquely each one of the copies of DNA that has come from the patient. And hopefully that would allow us to lower the limit of detection already. So that's our hope. But there is always a limitation with working with cell-free DNA, that the number of copies of the mutated allele, let's say, just not present enough for us to detect. Yeah.
Thank you. Other questions? I think we've got some online.
Yeah, I think this is for you, Ben. Do you also plan to look at methylation patterns on long read in individual cases?
Yeah, that's something we're definitely looking at. We are basically due to receive a sort of higher output sequencer anytime soon. So we should be able to get more data out of that. And yeah, one of the benefits of the specific sort of sequencing that I'm looking at, which is nanopore sequencing. I didn't go into it because I could spend another 5 minutes on that. But there's no amplification or anything, so the methylation status is maintained. So there's already been some papers out there that have shown we can use it for the diagnosis of certain methylation disorders like Prader-Willi, Angelman syndrome and stuff like that. So yeah, definitely something we're looking at in the future. Thanks.
Other questions? David, I know there are some online.
I think they've all been covered, though. The only one that hasn't been covered is, will you be publishing soon, Flavia?
I hope so, but we don't know.
Other questions? Maybe Amy, a question for you. I know this is often a huge collaborative effort, often internationally, and I was at the CHIP retreat recently. I wonder if you can say a bit about that and the importance of collaboration in this space.
Yeah, absolutely. So as you saw from the slide, it's enabled us to generate this very large kind of single disease cohort of children which is quite unique. And the collaboration has been really effective in that we've all been able to learn from each other. We're able to share techniques. Although we're working on data sharing, which is always challenging, or at least, for example, we've got a reanalysis project, which Maria's been involved in, where we've been able to apply the same techniques, and we've got about an 8% uplift just on short read sequencing from a very quick reanalysis. We're all learning from each other about long read sequencing. So the interpretation of long read sequencing is very challenging because there are not very good control cohorts published of long read sequencing data sets. So we're sharing expertise in that way. And I think the main thing is really having that international impetus to push things forward, also in terms of moving into the next stage, which we hope will be novel therapies for these children. And then we should also say that the vision of IPCHIP, which Gosh is involved with, is to now bring in other rare disease cohorts. So gemstones, which is neonatal hypertonia or infantile hypertonia, will be the next cohort, and we're hoping to bring on other rare disease cohorts from Gosh into this same kind of model of international collaboration and kind of precision child health.
Thank you. No, yeah, you could absolutely see that as part of the retreat and the way that people were interacting. Thank you. Other questions?
Flavia, another one for you. Do blood-derived DNA fragment truly reflect neuronal mosaicism in focal epilepsy?
Sorry, could you repeat that for me, please?
Do blood-derived DNA fragment truly reflect neuronal mosaicism in focal epilepsy?
No, not really. So the experience we've had in the past is that with this specific type of disease, because of this mosaicism that I explained during the presentation, it's very variable, the percentage of patients in which we find the mutation also present in blood. For example, out of the 11 that we've detected changes in the DNA, only one of the patients has we've also been able to find the change in the DNA in the blood. So I would say if we were to test DNA from blood, we would be missing the vast majority of the diagnosis.
Thank you. Any other questions? No? No? I think we'll end there. Thank you. Really great session.